Namaste students, Krishna Vidya, I JVWU Dr. Biochemistry. In this session, we are going to discuss about the basic concept of the genomics and proteomics. Okay? So first of all, let's see what are the concepts of genetics. So in the last classes, we have already discussed about the genetics based with the molecular structure and function of the genetic behavior. It is concerned with the cell and of the organ also. Right? So here you can see the basic when we see the cell is dominance or the cell is having a different pattern of inheritance from the parents or the offspring as the genetic distribution of the variations and the change in the population. So, yeah. So here you can see different when we see the genomics is the study of the complete set of DNA. Okay, that means including all the genes in a person or other organisms. Okay. So genomics provides an overall the complete set of the genetic instruction provided by the DNA. So while transcriptomics look into the gene expression and the pattern, the proteomics studies or the dynamic protein products and their interaction via the metabolic or other intermediate steps, understanding the organisms or entire metabolism. So genomics, when we see, it is the study which refers to the complete set of a gene or a genetic material that is present in a cell or an organism. So that means the proteomics is a branch of a molecular biology that studies the set of the protein expressed by the genomes of an organism. So why is it so important to study both genomes and proteomics? Proteomics complement here genomics, complement the genomics and is useful for scientists who want to test their hypothesis with the based on the gene. Okay? Even though all the cells of the multicellular organism depend on a gene expression. Okay? So what are the genes? So here why it is important? Gaining a better understanding of interaction between the gene and the environment by the means of genomics and it helps the researchers to find and improve the health and prevent the like diabetes or such as the modifying the diabetes or the exercise or planning to prevent or delay the onset of the type of 2 diabetes in the people who carries the genetic predisposition. Okay? So here the three main activity of proteomics we can see the basic technology like cornerstones that includes the method of fractionate complex proteins or a peptide mixture or the MS to acquire the data necessary to identify the individual proteins, okay, and bioinformatics to analyze and assemble the MS data. Okay. So here, what is the goal here? To generate a the goal of the proteomics here to monitor the properties of the entire complete complement of a protein from a given cell or the organism to determine how this properties changes or reverse response to various physiological states or such as signaling ligands or the cell cycle and the disease. So example, the here study of a complex diseases such as the health diseases or the asthma or cancer or having the like because of having the diseases typically caused by the combination of genetic or the environmental factors of an individual. Okay? So here, so how a proteomics is being done? There are multiple methods of study of the protein. Generally, maybe detection of the antibodies or immunoassay or a mass spectrometry. Okay, so Mark Wilkin termed the proteomes and proteomics and were coined in early 1990s. Mark Wilkins and a student of an Australian McGill University. Okay, in order to mirror the term genomics and the genome, which was represented as the entire collection of genes of the organism. So the functional protein aims in the biological function of a protein or a protein group or a class of protein. With the level that includes the characterization of the enzyme activities as well as the protein interaction or the post-translation modification of the protein. So goal here or in a structure protein is to determine the structure for the protein in a cells or organism or appropriate having several thousands of the protein structure being determined or deposited into the plasma or the gene bank. So here, this when the study of a nucleic acid begins and the discovery of the DNA process through the study of the gene in a small fragments and how it explores the field of genomics and the genomics study is the study of the genomes or includes the complete set of the genomes that include nucleotide sequences and the advantages of genomes that have been possible by the DNA sequence or the technology or mapping of the genes. So the genome mapping can be proceeded by finding the locations of the genes on which the each chromosome or a map can be created or compared. So physical maps into the intermediate details of the smaller region of the chromosome into the delayed part or a physical map, representation of the physical distance in a nucleotide or between the genes and the genetic markers. So both the genetic linkage map and the physical map are required to build up the complete picture of the genomes and have a complete map of the genomes made for the researcher to study the individual genes. The human genomes help in search of being the diverse to identify the human diseases caused the gene related to the illness such as the cancer, heart diseases, or the cystic fibrosis, or to name a few conditions, or the gene maps provide the outlines of the physical maps or to provide the detail. It's easy to understand why both the type of the genome mapping techniques are important. The big pictures or the information of the obtained to each other. Okay. So the gene mapping is still on the ongoing process and for advanced techniques are developed and more advanced are expected. So genome mapping is system complete complicated puzzles using every piece or available data or the mapping of the information generated in the laboratories over the world and entire into a cell central database or such as the National Center of the Biotechnology or Information. So applying the genomics, the introduction of the genome or DNA sequence and the whole genome sequence project, particularly the human genome project, was expanded in applicability of the JDNA sequence of the information. So the genomics is now being used as a wide range of variety of the fields such as the metagenomics or the pharmacogenomics or the mitochondrial genomics. So the most commonly used particles is the uh, yeah, the genomics in a like understanding or find the for disease or predicting the disease risk of an individual, genome-wide associated studies or pharmacogenetic or metagenome creation of a new biofuels, mitochondrial DNA, genomics of the mitochondrial interactions, genomics of the forensic sciences, which involves the information genomics in agriculture are being used, proteomics are being used. So applying genomics like mitochondrial genomics, the mitochondria are intercellular organelles that contains their own DNA. Okay? So mitochondrial DNA mutations is a rapid rate of often used in a study of evolutionary relationship. So certain features like see where in most multicellular organisms or the mitochondrial DNA passes on from the mother during the process of fertilization. For this reason, mitochondrial genomics are often used in the trace genealogy, where forensic is also used in the analysis of the forensic analysis. That means the information clues about the DNA sample found in a crime or a scene and found in a evidences in the court cases and certain genetic markers being used in the forensic analysis. So genomic analysis have been become useful in the field and in okay. So it may be a collaboration or effort between the academic research institutes or the forensic department. Yeah. Okay. So the powder, in fact, here the maybe the use of the microbial genomic or research determine the specific strains of and that's being used in the mailing eventually. Okay. So the genomics in agriculture being used to reduce the trial or and the failure involves in the scientific research of the certain extents of it, such as drought sensitive crops are more tolerant in the dry seasons or the proteomics where the proteins are the final products of the genes that pre-form the functions encoded by the genes or the proteins are being composed of the amino acid and plays an important role in the cell. So all enzymes except the proteins act as a catalyst and that affects the rate of the reactions or the proteins are all regulatory molecules and are hormones that transport the proteins such as the hemoglobin and help in the transport oxygen to various organs or the antibodies that depend or against the foreign particles are also being the protein or the protein of the entirely the set of the proteins produced by the cell types or the proteins can be studied using the knowledge of the genomics or because of the gene code of the mRNA. So the genomics and the proteomics of a patient suffering from specific diseases are being studied to understand the genetic manifestations of the disease. So the most prominent disease being studied with the proteomics approaches is the cancer. The proteomics approaches is being used to improve the screening or early detection of the cancer. So this is achieved by identifying the protein whose expression is affected or by disease process or the individual proteins being called as the biomarker. Okay. So here the study proteomics is the study of entire the protein expression given by a cell under the certain environmental condition in multicellular organism and different cell types being mutated. Proteomics means changing when the change in the environmental unlike the genomes or the proteomics is dynamic or constant fluxes there. They use the knowledge of the genome cell. Thank you. This session is powered by 2.0 with the Vidyapeth University. If you have any queries, we'll discuss in the